Three, two, one, go. Okay, everybody. I don't know about you, but I am like at the edge, right at the edge. Um, I like waking up in the middle of the night, not able to sleep. I think perhaps could be the election. I don't know about you. But like things are starting to slide, which is challenging. I hope it's not causing you all any problems. I'm just a little behind in grading. I also have great empathy slash compassion. Not sure what the proper term is for whatever you may be going through as coronavirus cases are multiplying throughout the country and still people are insisting that it's not a big deal and there's an election. Anyway, so, if you need a little extra time, please, 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 when you ask for that, what's very helpful is if you give me some specifics, not I need more time, like how much more time, what assignments, but say, these three assignments and then list them. I will have them done by Sunday night, the ones that are due on Thursday, or the Sunday night ones I can do by Tuesday of the following week, whatever. You tell me by when, and then I can go in and change those due dates to help you out, okay? Now, I also encourage you to do what you can to the best of your ability so that stuff just doesn't pile up for later. So you're not like giving yourself a yucky present in a couple weeks where you have a whole bunch of stuff that needs to get done before class is done. It's always that balance. So here we go. Week 11, it's week 11, oh my goodness, out of 15. So we're cruising, we're close to the end. And this section about DNA and the central dogma of biology, how DNA translates into proteins, specifically into amino acids that then link up to become proteins. When we cover this in high school biology, we spend like 6 weeks and still like, I don't know, 60% of the class ends up getting it and 40% still struggle. So if you are like, what are we supposed to do this week? Please don't feel like you're stupid. Please don't. It's challenging information and we just rush through it in a week. Oh, here you go. But hopefully this content helps you figure it out because it's kind of an amazing thing. I think it's one of those sort of miraculous, like biology is truly magic in many ways, things to know. So the specific information of how DNA actually translates into proteins. So the DNA, of course, stays in the cell. It gets translated. I hope I'm saying that right. Translated. I think it gets transcribed into a messenger RNA, which then leaves the cell, goes to a ribosome where it gets translated. I might be getting those backwards. We'll have to look later. Transcription is first. Yes, translation is later. And then it gets turned into an amino acid sequence, and then that folds up really fancy crazy to make a protein. Then this one goes a little bit into how mutations can happen, which they do regularly. Usually they're corrected. Many of them, mutations have no effect at all. Some can be quite devastating. So about mutations and how they happen and what they are. And then as a way to apply this information, we're learning about antibiotic resistance. So I know sometimes this class can sort of be like, oh, dude, why'd I ever take this? Like, I didn't want to know that. This is one of those subjects. If you weren't already worried about, decreasing water supplies, global climate change, all kinds of other stuff, here's another one to add to your plate. Antibiotic resistance because of overexposure to our amazing antibiotic medicines. Anybody who understands evolution could have predicted this. That a lot of the microbes that that are out there that cause us a lot of problems are now resistant to the medicines that we would use to deal with them. It's challenging. And then how does that happen? And then some information about typically not a problem, Staph aureus. It's on your skin right now. But if it gets in your bloodstream, you can die, causes infections. And if the infections get septic, it's a huge problem. So here we go. And also because there's a lot of MRSA, methicillin resistant. Methicillin is an antibiotic. Methicillin resistant Staph aureus, MRSA or MRSA, is becoming a big issue like in hospitals and other places. So do your patients have a resistant strain of Staph aureus? Let's hop right up there and check that out. Okay, so first off, you've got 2 patients, Alex and Betty. There's 2 genes that we're looking at. So I think they have a nasal swab to see if they have the resistant Staph aureus. There's two different resistant genes that we're looking at here. Gene 1 and gene 2. So this is their DNA sequence. So if you recall, DNA gets transcribed into messenger RNA by the complementarity rules. So C's go to G's, G's go to C's. So it's always CG and AT in DNA. C's and G's match up, and A's and T's match up. So if you have one strand of the DNA, you know what the other strand is because it's the complementary base pairs. Okay. Now, if we're going from a DNA into a messenger RNA, wherever you see an A, you would expect that there would be a T on the messenger RNA, except that the way that it just worked out, evolutionarily speaking, messenger RNAs, ribonucleic acids, RNAs, do not have any thymine. So there can't be a T that matches up with that A or adenine. Instead, it's a uracil, it's a U. So it's just something to remember, okay? So here we get a GC, yeah, that's where my brain goes, U, GCU, right? GGU, ACA, and on and on and on, okay? So first you have to transcribe the DNA into a messenger RNA sequence. There we go. Transcribe each DNA to its complementary RNA sequence. Remember, these are the pairing rules. A goes to U, T goes to A, da 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 da. Okay, so there we have an example, and then what the RNA sequence is. Now, I added this because I find this to be a lot easier. Like if we have a code that says AUG, A is the first one in the middle, right? U is the second ring, and then you find G right here, and look, it's methionine, or M. I like that one better, so I put it in there. But if that doesn't float your boat, or you used this one before and you prefer it, the first position A, then you find the second position up here, U, and so it's going to be somewhere in this box, and AUG is that one, and it's methionine or M. So either way. So once you get all of those there I go again, translated, right, into amino acids, ALA, GLY, through whatever, the abbreviated is AGTT, right? So that's his gene 1. Now, if the bacteria in Alex Torres's nostrils is susceptible to the antibiotic, then it will be AGTT-ERI, right? That means that antibiotic will be effective against the bacteria in his nose. If he has a K here instead of a T, that is the resistant form of the gene, and that antibiotic will not work. Fusidic acid will not work if that's what was in his DNA of the bacteria that's in his nose. Okay, so you have to go through the process of transcribing to a messenger RNA, to an RNA, and then translating that RNA into amino acid sequence, and then comparing for gene 1 and gene 2 whether or not they have the resistant strain, which is a problem, or the susceptible strain, which means that this antibiotic would be effective. Okay, and you have to do it for both of them. Got it? Okay. So the next thing that I thought would be challenging, let's just kind of see what else is there. So that was, do these patients have this? And then you check yourself, and then we check out the gene function worksheet right here. So this actually looks at the same kind of things. It requires that you already understand the process of transcription and translation, and mutations that can make bacteria resistant to antibiotics. So this is fusidic acid and ampicillin resistance, looking at where that happens in the cell, okay? Indicate where in this bacterial cell each of the 2 genes function. So they're all labeled like A, B, C, D. I think that's it, right? A, B, C, D. Yeah, I don't think there's an E there. So you can answer A. Oh, I'm glad that that's there, because last year there was some wacky answers there. What part of the bacterial cell fusidic acid would attack? So, you know, you can actually, like, then you would label it, or you can say, like, the cell membrane, or whatever the answer is. So hopefully this, you know, what happens when a bacteria is resistant to all antibiotics, you can answer that without even going over this stuff, and then it just applies it to the world in general. Okay, now back to here. And then, you know, the regular quiz, and then continue on. So here we have our peer review, and I believe this tells you what to do here. This is a new thing, so pardon me if I'm not super helpful because I'm also figuring it out. I did get one email of someone going, but I don't see where I'm supposed to do the peer review, but this was like Sunday before it was technically due. So now that this assignment has been due, everybody has 2 people that are assigned. You're assigned 2 people to do a peer review. If you handed it in on time, if you didn't submit it on time, then it didn't automatically assign you anything. And I think I went in and assigned things to people, but if you don't see anything there, please let me know, because I'll have to go in and give you an assignment, give you 2 people to peer review. Okay, so it tells you where to find that, your to-do list. It tells you what to do and the 2 people or whatever, right? Specific things, yeah, there we go. Yep, contact me, yep, indeed. Specific things to look at and comment on. So often in these kind of peer critique, students are like, "Oh, great job," and like that's it, or "Oh, you did it was good to read." But what's way better is if you point out like, "Oh, this is grammatically incorrect," or "I think this is a typo." Like if you are a good critical reader, then they can take your comments and actually improve, which is the point of doing the peer review. So I would highly recommend that. Okay, so I think I just got us out of the modules view too. Sorry. And I think we're just about done because I think that's also just about the end of the module. Oh, except like the big giant, not to be overwhelming or anything. This is going to move in just a bit. The final part of the coronavirus fact sheet thing. So I am endeavoring to grade your coronavirus in California. What are we doing here? The fact sheets that you've done so far from week 10, so that you have my feedback and then you have peer review before this is due on Friday. So this is like a little fact sheet that would be somewhere in a community area. I mean, little, whatever, like a trifold brochure, like, you know, at the dentist office, or stapled up on a telephone pole at the intersection or whatever. And it should have, like, here's a great example. So you can kind of model this sort of in how you're designing yours. So it's a lot of information, but it's pretty easy to read with bullet points. And then you need to have good quality references. You don't have to have this many, but it's good to have a lot of references. It makes it look like you did your due diligence, and all the specific things that need to be here. And then, indeed, yes, always look at the rubric. Here is the rubric. Please use it. 21 points. That's a lot of points. And I think the next one here gives you some advice on how to find images, because you need to find images that you are legally able to use, not just steal them off the internet kind of thing. That's not what we do anymore. So Flickr Creative Commons. So the Creative Commons, these are ones that are not owned by anyone and that you are free to use these images. And it's always still nice to cite where you got the image from. Anyway, I think that's about it. Just be kind to yourselves this week, please. It's a rough ride, man. This is unprecedented. I am so tired of using that word, but it continues to be unprecedented. So be gentle with yourselves, and please let me know if you have any concerns, thoughts, needs that I might be able to help with. Also, nobody's been showing up. Well, not nobody, but like one person in the last 3 weeks for the Wednesday Zoom session, which is also technically my office hours, but I'm still there if you need to ask about an assignment. It's a lot more private space now, so if you needed to talk about your grade, you know, I can, it's likely you'll be the only person on, so don't feel like it's this, you know, everybody in class is going to be on there. It's really time to have some private time with me if you need that. Okie doke.